This method can help us answer key questions in the CAR T cell and immune therapy field, such as further genetic modification of CAR T cells and combination with other immune therapies. The main advantage of this technique is it allows comparisons between the lymphoreplete and the lymphodepleted settings. This allows us to see the role of endogenous immune cells in both disease progression and their interaction with therapeutic CAR T cells.
The implication of this technique extends to therapy or diagnosis of solid tumor, because the lessons learned in this model can be directly translated to CAR T cells with other targets, such as CA.
To begin this procedure, first prepare all needed media and cells as outlined in the text protocol. On day 1, add complete DMEM to 15 square centimeter culture dishes. Seed 7.5 million platinum E cells in these dishes and incubate overnight at 37 degrees Celsius with 5% carbon dioxide. The next day, prepare 3 milliliters of transfection solution 2 for each dish as outlined in the text protocol. Vortex this solution for 10 seconds and then let it rest for 5 minutes. Remove the medium from the dishes and replace it with 12 milliliters of transfection solution 1. Add 3 milliliters of transfection solution 2 dropwise to each plate, making sure to perform the addition evenly across the plate. Rock the plates gently with a side-to-side.